Hey there, pre-med! In this video, we're going to talk about one of the most important topics on the MCAT, possibly the most high-yield topic you'll find when going through resources and practice exams, and that is your amino acids in biochemistry. There's a lot to know about our amino acids and a lot to conceptualize here, so it can be really overwhelming to both stay organized and make sure you're learning the concepts of the amino acids in the way that they're going to be tested on exam day. In this video, what we're going to do is make you a study guide, including everything you could possibly need to know about the individual amino acids that will be tested on the MCAT and all of their organic and biochemical characteristics that define their function. Of course, amino acids don't work in isolation, they work in concert with other amino acids in proteins, and so that'll be covered in a separate video getting into the different protein levels, primary, secondary, tertiary, and quaternary. In this video, we're going to focus on free amino acids and their characteristics. Before we get started on the differences between the amino acids we need to know for test day, let's talk about their general features. All of our proteogenic alpha amino acids have this general structure, which is a central carbon bound on one side to an amino group, so in this case NH2, and on the other side to a carboxylic acid group, CO2H. That central carbon then is also bound to a hydrogen and an R group. An R group is just a placeholder. Each amino acid has a unique R group, and that's how we're going to differentiate them in this video. Now, with the exception of one amino acid, all of our alpha amino acids are chiral, which means there are 4 unique substituents around this central carbon. That's an important feature as the isomerization and stereospecificity of chiral amino acids can impact their function. Another important note is that in the body, the amino group is going to be positively charged. So it's going to be NH3+. This is because amino groups act as bases that accept protons from their environment. On the other side, the carboxylic acid will be deprotonated at physiological pH because carboxylic acids are acids, which means they donate hydrogens to the solution. So we can see here that we have a negative charge on one side, a positive charge on the other, which means that the overall molecule is net neutral, unless there's something happening with the R group. And we call this a Zwitterion, where we have ionic character in the molecule, but they net out to be a neutral character overall. We're going to use this general structure of our central carbon, amino group, carboxy group, and R group as we move through each amino acid that you need to know for test day. It's important to recognize that this is the backbone that stays consistent, and the unique features are what we're going to discuss in the R group. Let's go ahead and build our study guide now. One of the trickiest parts of amino acids is that they have different categories depending on what we care about what's going on in the protein or their interactions with each other. In general, though, there is a binary choice where all amino acids will either be hydrophobic or hydrophilic. This ties into their polarity, where amino acids will either be nonpolar or polar, and on the far side of polar is charged. Now, nonpolar and hydrophobic means that they don't interact well with water. Nonpolar in organic chemistry means that we don't have strong dipole moments between our atoms in bonds, and those dipole moments are created by differences in electronegativity. If we don't have big differences in electronegativity, we're considered nonpolar. In our aqueous environment in our bodies, that also usually means hydrophobic because we can't interact well with the polar molecule that is water. Like with like is very common in this setting, and so a hydrophobic molecule will not be able to have dipole-dipole interactions or hydrogen bonds with water, creating those intermolecular forces for them to interact. So they're going to kind of want to stay away from each other. We have nothing in common, we have nothing to interact with, therefore we're hydrophobic, disliking water. Let's start there, and we're going to work through our hydrophobic nonpolar amino acids and move our way through to the polar side. Our first 4 amino acids that we're going to look at are alanine, valine, leucine, and isoleucine. Let's zoom in and take a closer look. So why are we looking at alanine, valine, leucine, and isoleucine all together? Well, let's take a look at their R groups, which I've drawn in a darker black color here. All 4 of their R groups are just hydrocarbons, right? They're just carbons and hydrogens, either in a chain or a single methyl group. And these hydrocarbon chains are called aliphatic groups in organic chemistry. So we'll go ahead and categorize our first subgroup called aliphatic. And this just means we have carbon chains that are non-aromatic rings. So Alanine just has a methyl group here. Valine has 3 carbons in kind of a V shape, kind of nice because it matches its name. And then leucine and isoleucine each have 4 carbons each. It's just that one goes straight chain and then a V for leucine, and then isoleucine kind of creates this longer sort of I chain, IL chain. Now you might think to yourself, I see exactly the same number of carbons and hydrogens between leucine and isoleucine, right? And they kind of have the same name. That's because these guys are isomers, specifically structural isomers. Isomers have the same molecular formula, the same number of atoms, but a different arrangement of those atoms. So leucine and isoleucine are structural isomers, which means they do have a different structure, they have different bonding, and they'll have a different IUPAC name, but the same molecular formula. So these guys are very similar, and they're all usually interchangeable in the sense that if we have one versus the other, it probably won't impact the performance of that enzyme or protein too much. The exception here is if the protein relies on branching. If the protein relies on branching, valine, leucine, and isoleucine will all behave similarly, but alanine with only one methyl group won't have that same branching feature. Branching can impact the surface area and how the amino acids interact with each other, so this could be relevant depending on what the question asks. Otherwise, all these are very nonpolar, very hydrophobic. They don't like interacting with water. You do need to know their full names, their single letters, and their 3 letters that are in parentheses, and of course, their R groups. Now, one additional note before we move on to other amino acids here, I do want to point out why I drew the amino acids in a specific way. This is because all of our amino acids are what we call L amino acids. And that means that with the exception of one special amino acid, they're all going to have the S configuration of chirality. Now, chirality is an organic chemistry concept. It's not deeply associated with this video, but I briefly want to show you why this is S or L amino acids. So we look here, nitrogen is going to have our first priority off of our chiral carbon because it has the highest atomic number. From there, we've got a tie between these 2 carbons, so we have to go one more atom out, and we can see that oxygens beat hydrogens in terms of atomic number. So this guy's going to get position number 2, and the methyl group will get position number 3. The hydrogen is in the back, which is very important for chirality because we can only establish the direction correctly if we know that the hydrogen is in the back. So then what we do is we draw an arrow from 1 to 2 to 3 priority, and we see that it is counterclockwise. Counterclockwise indicates S configuration. And in the world of amino acids, in a lot of macromolecules, we substitute an older designation, which is called L or D. This is our L amino acid because, again, it's in the S configuration, and our nitrogen here is on the left. There's one exception to that rule, which we will see, and I will point out when we get there. All right, so we had our first 4 amino acids. Let's get 4 more. We're going to start with our next one, methionine. So let's take a look at methionine. Methionine has 2 carbons and then a sulfur and another carbon. This functional group in organic chemistry is called a thioether. As in ether, ethers are normally single bonded oxygens between 2 carbons. Thio is our prefix for sulfur-based compounds. So a sulfur-based ether, essentially. Now, you may be thinking, hey, Amanda, you put this in the very nonpolar category, but sulfur, isn't it kind of like oxygen? Isn't it a polar atom? It's got 6 valence electrons. And I know you may think that, but remember, when it comes to polarity, we're talking about dipole moments, and dipole moments are produced due to a difference in electronegativity. And what's interesting is that sulfur, by being 1 row down, actually has almost exactly the same electronegativity as carbon, around 2.5. So these guys are considered incredibly nonpolar bonds. So methionine is another nonpolar hydrophobic amino acid. It just has a thioether. It's got another atom instead of carbon, but with a similar electronegativity. Now, a couple other fun facts about methionine. Methionine is coded for by the start codon AUG. AUG is our mRNA sequence that codes for the very first amino acid in a primary chain that's translated by a ribosome. So it's a little genetics connection. So this start codon, which usually kicks off our chain of amino acids that's being formed by a translation, codes for methionine. So the tRNA that is complementary to AUG is the tRNA that holds onto methionine. So we'll often see that methionine is our very first amino acid in a primary sequence. Okay, we did methionine. Now let's move over to our next set of amino acids, which includes our next subgroup. And this subgroup is our aromatics. Aromatics is another organic term that is referring to a planar ring structure that has aromaticity, which is also known as conjugation, where we have alternating single and double bonds that can move. So I've drawn them as double bonds being here and here and here, but in reality, they can shift positions due to a delocalization of electrons. This again is a very organic chemistry concept, and we're just using the naming convention here to describe this category, which are these 3 amino acids: phenylalanine, tyrosine, and tryptophan. Now, what's important here is that even though these are all aromatics, they do have really different features in each of their R groups. So they all are aromatic rings, which means if we're talking about aromaticity, they can be interchangeable, but sometimes they can have some different behaviors as well. So let's zoom in a little bit more and talk about phenylalanine. Okay, so phenylalanine, I kind of love it because its name describes its organic molecule. A phenyl group is a 6-membered, 6-carbon ring that's derived from benzene. So I'm going to sketch benzene for you here. So this is a full benzene ring. A phenyl ring is just a benzene that's bound to something. So it has one less hydrogen, right? So instead of a hydrogen here, it's bound to this carbon. That's the only difference between a phenyl ring like we have here and a benzene ring. And alanine, which you may remember, we can zoom over to alanine here, has just a single carbon. And so you can see here, oh yeah, single carbon plus a ring, phenylalanine. So love that, nice clean naming here. And it's great practice for our ring-based structures. You can look at this thing and tell me, right, it's very nonpolar. There is a lot of balanced delocalized electrons, no real dipole moments here, no functional groups that can hydrogen bond. So this thing's going to be quite nonpolar, hydrophobic, stay away from water. Let's go down now to tryptophan. So tryptophan is a weird looking one, right? It's our chunkiest ring that we have. This type of ring is called an indole ring, I-N-D-O-L-E. And we can see here that it's got a nitrogen, but it's also got an aromatic ring structure. So tryptophan is a little bit of a unique situation where nitrogens can typically hydrogen bond, right? Nitrogens, oxygens, and fluorines. But because of the real nonpolar character of this indole ring and the delocalization of electrons, it's not really going to hydrogen bond. And so this tryptophan still counts as our nonpolar amino acid. So characterized in biochemistry, this is a nonpolar amino acid because it's just so unlikely to interact in a meaningful way with water. It's more likely to hide from water. Now to break down this organic chemistry just a bit more, an indole ring is a benzene ring, which we have here, right, our 6-membered carbon ring, plus what we call a pyrrole ring. All right, a pyrrole ring here with our nitrogen and 4 carbons with double bonds. So we have a nitrogen pyrrole ring plus a benzene ring. That's going to equal an indole ring. If you see any of these words on the MCAT coming up for organic chemistry, tryptophan is a good example here. So you may have heard the myth that there's tons of tryptophan in turkey and that's why you get sleepy after Thanksgiving. And there is some truth to the fact that tryptophan does help with making you feel good with serotonin and make you feel sleepy with melatonin. But turkey doesn't have meaningfully more tryptophan than any other protein. It's more likely you just ate a lot of food and got sleepy after that. Still, it's a great way to think about the importance of tryptophan in our diet. Okay, let's do our final aromatic, and this is where things are going to get a little funky, so stay with me. So I'm going to zoom out just a bit to show you that tyrosine has actually crossed us over into the polar category. And this is absolutely true because tyrosine has what we call a phenol group. And a phenol group, as you can see, has a hydroxy group here. And this hydroxy group, this OH, separate from our ring structure, even though the ring structure is quite stabilizing, this hydroxy group is going to be able to participate in hydrogen bonds, which means it wants to hang out with water because water can also hydrogen bond, right? It has something more similar than all the rest of the molecules we've talked about so far to water. Now, this may be a little confusing depending on the resource you've looked at. And I want to just slow down just for a second to really clarify what we mean by polar and nonpolar. In organic chemistry, Phenol groups, especially when they're bound to other carbons, are often actually seen as nonpolar. They have a really balanced delocalization of electrons. But in biochemistry, what we really care about is how these molecules behave in aqueous environments like water in our bodies, essentially. It's not about these things acting in a lab and measuring their polarity in isolation. It's about using them in our bodies, in our proteins, in our enzymes. And tyrosine, despite being pretty nonpolar, if we're just measuring the polarity, is going to act in such a way that it can interact with water because of its phenol group and its OH here on the end. So that's why we put it as polar, even though some resources you may look at, particularly if they're organic chemistry, may label tyrosine as more nonpolar. I just want you to know for the MCAT's purposes, we take the biochemistry definition, which means that these guys are polar. It can also get deprotonated if it's in a basic environment, which would make it even more polar because it'll have a negative charge and can interact with water even further. So tyrosine is a little bit unique. Even though it is aromatic, it counts as polar, whereas phenylalanine and tryptophan are nonpolar and aromatic. And again, this is where the subcategories to the overall categories become important for you to stay organized and making sure that you're saying, what am I describing? Aromatic is just the structure. Polar and nonpolar is how it's interacting with water. Some other fun facts about tyrosine. Tyrosine, much like tryptophan, is an incredibly important precursor for many neurotransmitters like dopamine. It also is the precursor for our T3 and T4 hormones produced by the thyroid. So they help regulate metabolism. If our tyrosine levels are low or too high, it's going to impact our ability to regulate our metabolism through thyroid action. So a lot of these precursors will show up in endocrine discussions because even though they're amino acids and they're about proteins, they also have individual actions in our signaling molecules. So it's a nice little overlap here, particularly with our aromatic amino acids. We've now done our first 8 amino acids. We've got 12 to go. We have a couple more that fall in the nonpolar region before we fully move over into the polar category. Let's talk about those two, which are proline and glycine. So proline, take a look at this weirdo. All right, proline has a ring structure as part of its backbone. So it does have an R group. The R group is just attached to our base amino group, and it forms this 5-membered ring with nitrogen in it. This ring, and I apologize to my organic chemistry aficionados if I'm pronouncing this wrong, is pyrrolidine. All right, again, the pyrrole for the 5-membered ring with nitrogen in it here, and it's part of our main chain. And the reason why this is so interesting and unique is because our peptide bonds that form our amino acid chains to make proteins form between our base chain nitrogen and our base chain carboxy groups. And so this is going to create a real funky kink in our straight line, or typically straight line of our amino acid. So proline is unique. It doesn't really have any other buddies in its own subcategory. It's kind of special, and it does have important ramifications in including proline in things like our secondary structure. Because it will cause these kind of rotations of our primary structure, it'll end up causing turns in the secondary structure as we start to fold the protein together, particularly in our structures called beta pleated sheets. You're also unlikely to find proline in the alpha helical substructures because, again, due to the fact that it causes this like kink and turn in our primary structure, it's not going to help us create a nice clean alpha helical secondary protein structure. So keeping an eye out for proline, it has some special characteristics here. Again, much like our indole ring, even though it's got a nitrogen, which can hydrogen bond, due to the unique characteristics of its ring structure, it doesn't have a strong enough dipole moment to really interact with water effectively, so it's going to count as nonpolar. Again, this one's kind of unique. It's much like methionine. We have 2 other special amino acids that go in this category that don't have their own subcategory buddies. And the next one of those is glycine. Let's go ahead and talk about glycine. Glycine is our smallest amino acid because it's got just a hydrogen as its R group, which means it's going to be unique in several ways. The first being that it does not have a chiral center. The central carbon here is surrounded by 2 hydrogens, and in order to be chiral, we need to have 4 unique substituents. So from an organic chemistry perspective, glycine is achiral. No chiral center. It's the only one out of our 20 proteogenic amino acids that has this characteristic. So I did draw it with the wedge dash diagram just for consistency's sake, but it does not have chirality. I'm going to gently take our polar line here from tyrosine and bring it down and gently include glycine in the nonpolar category, because generally speaking, when we're looking at resources for the MCAT, they consider glycine to be nonpolar. But I want you to know that you can still find glycine in polar areas of the protein. It will still be there, it'll be able to hang out in both spots, kind of can do whatever because it's so small, it's like, yeah, sure, you can snuggle in with us. So glycine is unique in that way. Even though it's nonpolar categorically, it can still hang out with the polar guys if needed. Okay, we zoom back out for a second. You can see that we've completed our first 10 of 20 amino acids. Before we dive into our polar and our charged amino acids to finish out this study guide, I'm Amanda Brem, and I've been coaching pre-meds on their MCAT journey since 2019. Please remember to subscribe to this channel for more videos on MCAT style content, test taking strategies, and mental fitness tips to help you perform your best on test day. And if you'd like to join me for my next live online small cohort style class, please go ahead and check out our next available courses in the link in the caption below. All right, let's dive back in and see our next players on the polar side of our amino acid study guide. All right, our next two are serine and threonine, and these are two of our biggest players on the MCAT, and let me show you why. So zooming in here, we can see that both serine and threonine have hydroxy groups. So that's definitely what makes them polar, because hydroxy groups are able to hydrogen bond with water. They're happy to hang out with water. And in serine, we can see that it's just a hydroxy group bound to a carbon. And in our threonine, we can see that we have a carbon and then another carbon, and then kind of teed up, threonine T, we have our hydroxy group here. Now, one interesting thing to note about the structure of threonine is we've defined chiral center as a carbon with 4 unique substituents, right? With our chiral carbon here. Well, this carbon, 1, 2, 3, 4 unique substituents. So this is our only amino acid with a second chiral center. It's just a fun feature there. Again, we usually are focused on still our, what we call our alpha carbon, which is our chiral carbon in the center here. By the way, the alpha is just referring to the fact that it's 1 carbon away from our highest priority substituent, which is our carboxylic acid. So we can have beta and gamma amino acids where it's multiple carbons away from our carboxylic acid groups, but that's not proteogenic amino acids. So our alpha amino acids are always what we're going to be paying attention to. Just recognize there could be other structures that are beta or gamma. Those are usually our neurotransmitter-based amino acids and not our proteogenic amino acids. Okay, so what makes threonine and serine such important players besides the fact that they're nice and polar? Well, in addition to hydrogen bonding with water, these hydroxy groups can be phosphorylated. They can actually bond with a phosphate group by removing the hydrogen and creating a bond between the oxygen and the phosphorus of a phosphate group and be phosphorylated. That's really important because phosphorylation is one of our major post-translational modifications that we can use to regulate proteins and enzymes. So you may have heard of a receptor protein called serine threonine kinases, receptor kinases, and that's because kinases phosphorylate things. Serines and threonines can be phosphorylated, and so by adding phosphate groups to these residues, these amino acids on this protein, we can activate or inactivate different messenger signals. So phosphorylation is a major regulation process. We see it all the time in biochemistry mechanisms, and so it's likely to show up on test day. Now there's one other amino acid that we just talked about that also has a phenol group, right? And that's our friend tyrosine. Why do you think I snuggled these guys up together? So let's go ahead and put a box around tyrosine because this is a secondary subgroup, and that subgroup is amino acids that can be phosphorylated. So if the passage asks you, they say, hey, this serine residue, its function in the enzyme was to be phosphorylated, but if we were to substitute it, which ones could we substitute in? And it would probably do the same thing. It would kind of be like a good substitute, same job. Threonine or tyrosine are going to be your best choices here. Pretty cool, right? Tyrosine's got a lot of overlap here, and we can again see it all comes down to the organic structure. Structure defines function. Now our next player is another contender for the most tested spot, and that is cysteine. Cysteine's another one of our special amino acids that doesn't really have a subgroup because it's so unique. Let's zoom into the structure and see why. So cysteine has an R group with a carbon group and then a sulfur with a hydrogen. This kind of looks like an OH, right? Except for that it's got a sulfur. So instead of hydroxy or alcohol, it's a thiol. Thio, again, for our sulfur, and then all as in alcohol for our SH, kind of like our OH. So sulfur and hydrogen, all right, this is definitely going to create a dipole moment. So even though it's not going to hydrogen bond, because only oxygen, nitrogen, and fluorine can hydrogen bond, the sulfur and hydrogen creates a big enough difference in electronegativity that will still have a dipole moment that we can work with here. So cysteine is definitely polar, and it also can do very, very cool things in our higher level protein structure. Particularly, it's involved in creating what's called a disulfide bond. Disulfide bonds are formed via oxidation. Oxidation is the loss of hydrogens when we're talking about oxidation in the world of organic chemistry. So what happens is that these sulfur hydrogen groups lose their hydrogens, oxidation, and when we have two sulfurs together that lose their hydrogens, they'll end up forming a covalent bond between the two sulfurs. So via oxidation, these guys, two sulfurs, two cysteines together, will form a disulfide bond or a disulfide bridge. These are covalent bonds, all right? These are strong covalent bonds, and they can only be broken by reducing conditions, by adding those hydrogens back in to cause them to break. This is going to have very important ramifications when we get to our quaternary structure, which is multiple subunits being held together, potentially by our disulfide bonds. And if you want to see how that may show up in biotechnology, please feel free to go check out my SDS page video, which really highlights the importance of these disulfide bonds in identifying different types of proteins and their structure. The other interesting thing about cysteine is that due to sulfur having a higher atomic number than oxygen, we actually have a different orientation around our chiral carbon. This is an R configuration, and it's still listed as L. All of our amino acids in our bodies, all the ones that we're going to work with, we always form L amino acids. But in cysteine's case, the L designation is associated with the R designation. L and R are completely different naming systems. L is referring to, again, our nitrogen group being in a specific location relative to the rest of the substituents. R is specifically naming the 4 unique functional groups and looking at them from a clockwise or counterclockwise direction. So cysteine is LR. All the rest of the amino acids are LS. So again, that's due to sulfur having a higher atomic number than oxygen. Okay, 7 to go. And our last 2 polar uncharged amino acids are asparagine and glutamine. Let's zoom in and take a look at their features. Okay, so asparagine and glutamine are great to learn together because they have very similar structures. We can see that asparagine has 1 carbon and then an amide group. We're talking organic chemistry, this is an amide. An amide has a double bonded oxygen to a carbon who's also bound to an amino group. And we can see that glutamine also has an amide group here. Asparagine, glutamine, a nitrogen plus a carbonyl. The only difference is that asparagine has 1 carbon and glutamine has 2. So that's how we differentiate. We look for the amide first, and then is there 1 carbon in between the amide and the chiral carbon, or 2? And if it's 1, it's A. If it's 2, it's G. I just remember that A is first in the alphabet. It has less carbons. G is later in the alphabet, has more carbon. Now, again, because A's and G's have already been used up with alanine and glycine, we get to some weird letters here. So we have N and Q for asparagine and glutamine. These often just take a decent amount of memorization, knowing they're a little different and a little special. Now, these amide groups do absolutely make these molecules polar because, again, nitrogen, when it has this nice free amine group here where it can move and interact, is going to act along with water and do hydrogen bonding. All right, so these guys can participate in hydrogen bonding, and these carbonyls can act as hydrogen bond acceptors. They don't have a hydrogen, but they can accept a hydrogen from water in a hydrogen bond. Reminder, hydrogen bonds are intermolecular forces. They're not full covalent bonds. It's kind of a misnomer, but they are very strong interactions that we see often in our molecules here. So asparagine and glutamine, again, have these nice amides, and they can participate in hydrogen bonding, which makes them nice and polar. We'll go ahead and put them in a gentle subcategory here because, again, these two are only two that have amides in their R groups. So we'll have amide-containing R groups as their kind of gentle special subgroup so that you know that those two kind of go together, very similar, kind of interchangeable. If we wanted to replace one with another, probably not much would change in terms of the protein's function. All right, zooming back out one more time, we have just 5 amino acids to go, and these are all now finally in our charged category. These are amino acids that have charges or can have charges on their R groups depending on their environment. We're going to start with our acidic or negatively charged amino acids, aspartate and glutamate. Let's zoom in. So aspartate and glutamate have similar names to the amino acids we just looked at, right? Asparagine and glutamine, and that's because they have almost exactly the same structures. The only difference is that aspartate and glutamate, instead of having an amine in this spot next to the carbonyl, they have another oxygen. They have another carboxylic acid. So instead of an amide, it's another carboxylic acid as their R group, but otherwise their structures are pretty much the same. Now, even though that's only one little teeny functional group difference, they have incredibly different features because carboxylic acids are negatively charged or deprotonated at physiological pH, much like our carboxylic acid in our main chain here, right? So while we add up and cancel out the positive charge of the amino group and the negative charge of the carboxy group, when we have another negative charge, these guys will end up with an overall charge of -1. So when we describe the charges of a protein, if we have glutamate or aspartate in there, it's going to be -1 for each of these amino acids at physiological pH. Now, these amino acids are characterized as acidic amino acids. And you might be surprised by that, because I know I was. Because I usually think of acidic compounds to being heavily protonated, right? They have lots of protons. They're usually positively charged, if anything, right? So what on earth are we doing here with a negative charge? Well, this is because these hydroxy groups have already acted as an acid. Acids donate protons, and this has already donated its proton, and it's now acting in its conjugate base form. Because again, once we've donated our protons, now we're going to be in our deprotonated or conjugate base form. So the reason these are described as acidic is because they've essentially already acted as an acid and now are in their conjugate base form. So we can go ahead and block these guys off as their subgroups. These are our only 2 acidic amino acids that we need to know for test day: aspartate and glutamate. The other way we would say that is aspartic acid and glutamic acid. The only difference there in terms of nomenclature, we use aspartic acid and glutamic acid when they're protonated, so when it would be like OH here, right? And we'll use aspartate and glutamate in its deprotonated form. ATE suffix is just the deprotonated form of an acid. Now, an interesting feature about aspartate and glutamate, I'm going to zoom out just for a second. Now, you may notice we talked about the serine and threonine and tyrosine as being able to be phosphorylated because it has a hydroxy group here that can bond with a phosphate group. And interestingly enough, so can the acidic amino acids. All right, so we're going to draw a little funky line here, drawing that they can also be phosphorylated. The difference is they don't form a true phosphate group because they have a carbonyl here, so it's a slightly different functional group. So the MCAT will call this phosphomimetic, meaning it can still be phosphorylated. It'll still allow the protein to do its job if we substitute aspartate in for serine, but it's not going to quite have the exact same effect as it would for serine, threonine, or tyrosine. So why is that important? Because once in a while, serine, threonine, or tyrosine will get substituted. Right? They're supposed to be phosphorylated and they got mutated. We substituted a different amino acid in. And they'll ask you, hey, which amino acid would allow us to retain the same effect? Will still be phosphorylated. And maybe serine, threonine, and tyrosine are not options. Okay, if they're not options, then and only then can you choose aspartate or glutamate as your backup options. So options 1, serine, threonine, or tyrosine. I remember that as thank yous because their single letter are TYS, thank yous. And then if these aren't available for phosphorylation, then you can go to D and E, aspartate and glutamate, as a secondary option for phosphorylation. And this just comes up a lot on the MCAT, so it's important to have this in your notes, that this is an available answer choice. And it's not about them being charged versus polar, it's about their ability to be phosphorylated. Okay, we're almost there. We have just one group left, and that is our basic amino acids. Let's work through them one by one. We're snuggling them in here. We have first lysine and arginine, and then we'll finally talk about histidine. All three of these, lysine, arginine, and histidine, are all basic amino acids. They all have kind of interesting different features though. Lysine, you'll notice, has this long kind of bent chain of 4 carbons, 1, 2, 3, 4, ending in an amino group. We know our amino groups. We have one in our base chain as well. So this is a classic amino group. And just like we just talked about with the acidic amino acids, our amino group is a basic functional group. It's going to accept protons from its environment at physiological pH. So this amino group will confer a +1 charge to lysine at physiological pH. So if we have lysine or K, it's going to be +1. And again, it's because it's now in its conjugate acid form, right? Because a base accepts protons, and once it does so, it's in its conjugate acid form. It's technically a conjugate acid. Now, arginine kind of has a funky structure too, right? It's got 1, 2, 3, and then a nitrogen, and then a carbon again, and then it's double bound to a nitrogen plus another amino group here. So we definitely end up, we have an NH2 plus though, because of the double bond here. It can transfer over to this other amino group. It's definitely got the +1, and from an organic functional group perspective, this is called a guanidine group. Again, apologies if I'm mispronouncing that. And then again, this subgroup here is an imine group because a double bonded nitrogen and carbon is an imine. But this overall group is called a guanidine group. So we have a primary amino group on lysine and end kind of terminal amino group on lysine. We've got this funky guanidine group for arginine. Both of them will be positively charged conjugate acid forms at physiological pH. They're long, they're chunky, they have very important functions, again, when it comes to regulation of proteins, because their charges can interact with other charges. We most often see this in our histone molecules in our DNA. So the histones are proteins that DNA is wrapped around. Both lysine and arginine are positively charged, and DNA, due to the phosphate groups, is negatively charged. So lysine and arginine will interact with DNA as residues on histone proteins to hold them tight and wrap them around and condense them down in our histone protein form of chromatin. So the more lysine and arginine residues we have, the tighter we'll hold the DNA, and the more heterochromatin we'll have. If you'd like to check out a more detailed video about the regulation of histone modifications and how that impacts our DNA gene expression, feel free to check out my histone modification video because that connection happens there, and it's a really fun and cool way of establishing the importance of lysine and arginine structure. All right, our final amino acid is our most complicated amino acid, in my opinion, and that is histidine. Histidine's down here on the bottom, and histidine is a special dude. Just like lysine and arginine, it is basic. It's got the amino groups here, but it's got the amino groups in this funky structure called an imidazole ring. And the imidazole ring, even though it can act as a base, it can accept hydrogens, because of its resonance structure, because of the way it's oriented in the ring, it's going to be less likely to act as a base than arginine and lysine. In fact, histidine's pI, which we'll talk about in a second, is about 7.8. It's very close to physiological pH, which means unlike its fellow basic amino acids, histidine is largely going to be neutral at physiological pH. Now, this is important because it can still act as a base. It's characterized as a base because it can accept protons, and it will do so as soon as the pH allows it to do so. But in our bodies, in our normal pH of 7.4, histidine is going to be largely uncharged. There's always a range with these things. There are some molecules of histidine that are probably charged, and it's easier for histidine to get a positive charge than any of the other non-basic amino acids. But I want you to know, for MCAT purposes, it's neutral, where arginine and lysine are positively charged at physiological pH. I am planning another video on PIs and how to calculate this alongside our different primary and secondary structures. So feel free to watch that video when it comes out to really kind of solidify this idea of why histidine is neutral. For right now, you can just make sure it's in your notes that it does not have a charge at physiological pH. Histidine does come up a lot on the MCAT in several different ways, partially because its unique imidazole ring allows it to do some cool binding properties. And that's particularly around binding metal cations. So these imidazole rings are able to be both electron donors and electron acceptors. And that's very important when we're talking about creating bonds, because bonds are an exchange or sharing of electrons. So what histidine can do is it can coordinate with metal cations like iron plus and magnesium plus 2+, these divalent cations. And what it can do is it can create these types of bonds called coordinate covalent bonds. And these types of bonds allow the sharing of electrons like regular covalent bonds, but in such a way that it allows the metal cation to retain its positive charge. This is essential in many enzymes where these cations act as the cofactor in the active site to bind to our molecule of interest. Like iron is the cofactor in the heme group in hemoglobin that allows oxygen to bind to hemoglobin. Without iron being able to retain its positive charge, it wouldn't be able to interact with oxygen. We wouldn't be able to carry it through our blood. And histidine is the residue on the enzyme that holds that cofactor in place, kind of like bracing an important feature so that it can then go bind to something else. So we'll often find histidine residues in the active sites of proteins that coordinate with positive cations as their cofactors. Similarly, histidine tags are used to bind to nickel-nickel chromatography. Nickel-nickel chromatography does kind of the same thing we just talked about, where we have nickel cations and we coat the resin beads of a column with nickel, and the nickel will interact with the histidine molecules. So if we wanted to make sure our molecule of interest bound to our column, bound to our stationary phase, what we would do is we would tag it with histidine or imidazole rings, tag it with histidine, make sure there's lots of histidines on that molecule of interest. It will then coordinate with nickel on the resin beads, bind to it, and allow us to elute out everything else and isolate our protein of interest, our molecule of interest, due to the histidine tags that we stuck on there in our lab technique. We'll then usually use just clean imidazole rings that are not attached to the amino acid to buffer out our molecule of interest and get it to release from nickel-nickel and get our protein of interest or our molecule of interest eluted out. So this is, again, a nice connection to column chromatography. It's a connection to enzymes and cofactors. So histidine does come up a lot. It's a very unique molecule, but it's really important for binding. And again, even though it's basic, it does not have a charge at physiological pH. Let's go ahead and circle up our basic amino acids and finish our chart. Okay, look at our full, beautiful chart of amino acids with our overlapping subcategories. You can really visualize what's going on. We've got their structures, we've got their full names, we've got their single letters, we've got their triple letters, we've got all their different organic structures in purple. We've got some fun features in different colors to indicate their special characteristics. We understand the relationship between the general categories of hydrophobic, hydrophilic, nonpolar, polar, and charged, and where we'll find each of these amino acids. We're going to go ahead and scooch up to the top of our study guide just to write some key core rules that we know are going to be important for test day, and then we'll wrap it up. Okay, rule number 1, all amino acids except for glycine are chiral, and that means that they have a carbon with 4 unique substituents. All of them are also L amino acids, and that means that for all except for cysteine, they are S, and cysteine is R. All of them are enantiomers, meaning they only have 2 forms except for threonine, which has 2 chiral centers and therefore can be diastereomeric, which means that they have more than 2 potential isomeric forms. Second thing, all amino acids have primary amino groups that are positively charged and primary carboxy groups that are negatively charged. This is except for proline, whose amino group is a secondary amino group because it's wrapped up in its ring. But these result in Zwitterions, where the amino group will be positively charged and the carboxy group will be negatively charged. Our third thing is that some of our amino acids we can't make in our body. In other words, they're essential in our diets. We need to consume them in order to use them for proteins. Those 9 essential amino acids are leucine, isoleucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, and histidine. Again, because histidine is extra special all the time, histidine can be produced by some adults, but it's still considered essential. The other 11 are all considered non-essential. In other words, we can make them in our bodies. Finally, all of these are what we call our proteogenic amino acids, which means we use them to click together to make proteins, our primary structure, and we do that via translation, which is our process of turning mRNA and translating that into proteins via ribosomes. This is an excellent connection to both genetics biology and cell biology, understanding where and when and how translation and development of these proteins happens from our free amino acids. Again, they have other functions that we talked about as well, but focusing on the protein structure and how these interact in protein structures is an important next step once you've learned these amino acids. Here, I've zoomed out one more time to show you our finished project with me pushed off to the side a little bit, so feel free to reference this, take a screenshot of this. But again, as always, I've talked about this in my other YouTube videos, I really encourage you to draw these out for yourself, make these for yourself, because the tactile experience of learning the structures and understanding the categorization is how you will memorize and get these right on test day. Amazing work. I know that was a hefty one, but this is important information that will definitely get you points on test day once you've gotten this down. I hope this was a great video for you. Please feel free to ask any questions in the comments. And as always, happy studying.